Feeling itchy? From bug bites to dryness, lots of things cause itchy skin, and recent research suggests the microbes on our skin may also play a role, highlighting an interplay between microbes and the itch-scratch cycle that could be useful for developing tactics to keep itch at bay. Welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM. Before we dive into today's video, please take a moment to fill out the survey linked in the description. It only takes a few minutes and helps us make better videos. We appreciate your support. Our skin is innervated by a network of neurons that sense stimuli and relay signals to the brain. Within that network is a special class of itch-provoking neurons called proreceptors. When something triggers proreceptors, the brain interprets the signal as "I'm itchy" and elicits a response. In this case, that response is scratching. Note this is different from how the brain interprets pain. Whereas pain causes us to withdraw from the stimulus, for example, quickly retracting your hand if you touch something hot, the itch sensation causes scratching. Scratching, then, is an evolutionarily conserved response that's meant to protect the skin by sloughing off the itch-causing agent and facilitating communication between the nervous system and the immune system to deal with the insult. And while scratching is a pleasurable sensation, it can also be destructive, particularly in skin conditions characterized by a chronic itch-scratch cycle. In atopic dermatitis, for instance, an overactive immune response in the skin causes inflammation that can cause dryness and trigger itching, rashes, and an increased risk for infection. As people scratch those itches, the skin gets more inflamed and itchier, and the cycle continues. Understanding this cycle would be valuable for figuring out how to break it. With that, research suggests that there is a microbial component of itch worth exploring. The skin is coated in a diverse range of microbes, and interactions between those microbes and the immune system promote skin barrier function. They can also trigger inflammation depending on the composition of the community. And there's a purported link between microbes on our skin and itchiness. Bacteria, in particular, may directly cause itch by interacting in some way with proreceptors, and/or indirectly by prompting release of immune molecules like histamines with known itch-inducing properties. Still, mechanistically, the intersection between skin bacteria and the itch-scratch response is still hazy, though recent research has offered insights. Those insights pertain to one particular bacterium, Staphylococcus aureus. Many people have this microbe on their skin without issue, though it can be pathogenic and is especially abundant in conditions like atopic dermatitis or eczema, where it can exacerbate disease. With that in mind, what role does S. aureus play in itch, if any? A 2023 study provided some answers. In this study, researchers applying S. aureus to the skin of mice resulted in more scratching behavior compared to control animals, as well as more skin damage from scratching. The researchers ruled out that the S. aureus-associated itching was caused by immune cells or factors known to mediate either itch or inflammation triggered by the microbe, such as mast cells, which produce histamines. They did, however, note that the bacterium localizes close to sensory nerves in the skin. The scientists systematically tested whether any of the known toxins or proteases the bacterium makes are responsible for itch, finding that none of them were, except for one. Mice infected with a strain of S. aureus lacking the gene for one protease, called V8, demonstrated less scratching and dermatitis. Injecting V8 alone into the cheeks of mice induced itch, as well as skin damage. Diving deeper into the mechanism, the scientists discovered that the V8 protease interacts with the receptor on proreceptive neurons, the ones that trigger itch. Specifically, the protease cleaves part of the receptor, which jumpstarts a signaling cascade that culminates in itch. It also activated human proreceptor neurons in vitro. Conditionally knocking down the gene encoding the receptor in mice led to less itching, as did pharmacologically blocking it with a drug targeting the receptor. These findings paint a detailed picture of a case where a skin bacterium directly induces itch and, in response, scratching. The authors suggest that hijacking the itch-scratch response may help the microbe gain access to deeper skin layers and disseminate in the body. Scratching could also potentially spread the bacterium to other hosts, though more research is needed on that front. The induction of scratching is important here, and that scratching seems to be involved in how the body responds to bacteria. Indeed, a recent study builds on the story by providing a molecular basis for scratching in the context of allergy and S. aureus colonization. In this study, scientists treated mice with allergens that caused an itchy response on their ears. Mice that were allowed to scratch their ears exhibited ear swelling and an influx of immune cells into the ear, whereas those that wore a collar preventing them from scratching did not, or at least not as much. This, coupled with additional experiments, suggested scratching elicits an inflammatory response. Notably, scratching triggered by this allergic response altered the skin microbiome, specifically reducing the abundance of certain bacteria, including Staphylococcus. These findings suggested that scratching may determine, in part, which microbes survive on the skin and controls pathogen growth through the induction of inflammation. To further explore this relationship, the scientists exposed the ears of mice to S. aureus for several days, and allowing an adaptive immune response to develop. They then exposed their ears to the bacterium again, after which the animals started scratching. As they scratched, they showed increased ear thickness and markers of inflammation. After one day, there was about a 10-fold increase in the concentration of S. aureus on the mouse ears. These parameters, that is, ear swelling, inflammation, and decreased bacterial load, were all reduced or lost entirely in animals that could not scratch or were missing a subset of itch-associated neurons. Now, one thing to note is that this study was conducted in the context of acute itch, not the chronic itch-scratch cycle like in atopic dermatitis, when incessant scratching can actually help S. aureus thrive. Those dynamics would need additional investigation. But it does indicate there is an immunological antibacterial basis to itching. Scratching can thus both help fight bacteria and perpetuate their growth depending on context. Moreover, there are a mix of bacterial factors like the V8 protease and neuroimmune pathways that drive the itch-scratch cycle. Now, we've talked a lot about S. aureus here, but it is likely there are other microbes and microbial factors that can similarly trigger itch. This doesn't mean just bacteria, but also fungi and viruses too. The more we tease apart those diverse interactions, the more we could leverage them for therapeutic means. For instance, developing topical pro or prebiotics designed to interrupt the itch-scratch cycle by modulating microbial composition and function on the skin. Even skin microbiota transplants could be an option. Ultimately, the more scientists scratch the itch to understand host and microbial underpinnings of itch, the more possibilities will arise. That's all for today. References for today's Microbial Minutes are in the description. If you enjoyed this video, be sure to subscribe to ASM's YouTube channel for more. As always, I want to thank you for listening, thank Ray Ortega for production, and I'll talk to you next time.